Like everybody from the lobby is in. All right, so good afternoon, everybody. Welcome to the June installment in the Hempseed Coalition's monthly research-driven webinar series. Cannot believe it is already June, already here. I'm your moderator for today, Christine Ely. As a reminder, if any of you are new to joining us, each month we feature live presentations from experts across the industry, followed by time for your questions and discussion. As a reminder, if you can't make it live to these presentations, we do record them, so you can always come and catch up whenever it fits your schedule. Before we kick things off with today's speaker, I want to invite Wendy Mosher, President and CEO of New West Genetics and Vice President of the Hempseed Coalition Advisory Board, to share a few words with you all today. Wendy?
Yeah, hi, thanks, Christine. Hi, everyone. I'm dating myself, but do you remember Romper Room with the Magic Mirror? I see familiar names. Thank you for coming, everyone. We appreciate it. Again, as Christine said, I serve as Vice President of the Board of Hempseed. I've been working on HFC since its inception in 2017, together with a number of other people. This grew out of an effort mandated by the Colorado state legislators to take a look at, you know, is hemp and feed a reasonable constituent? It was drew together multiple hemp industry stakeholders, but also key to getting this through was experts in feed, veterinarians, Cattlemen's Association, AFCO representatives, FDA. They were all a part of that 6-month study, and out of that study came Hempseed Coalition. So we are a 501c3 with a mission to increase feed nutrition options, sustainable feed production, as well as to increase the markets for hemp, of course. You may have heard it's essential in agriculture to valorize all parts of the plant. So if you are in fiber, this applies to you too. This impacts all of the stakeholders in the industry. If you're an animal feed mill or CPG brand, we're really glad you're here. This is an important opportunity for you to differentiate and meet your customers where they are. No brand can continue to ignore that their consumers are hungry to improve their own diets and their animals' diets, and to source that sustainably. It's no longer a luxury, right? So what exactly does HFC do? We directly submit applications to the regulators for the approval of the use of hemp grain in animal feed. And then we respond and iterate back and forth with them for any, additional needed trials, et cetera, to enable those applications. There were no approvals before we started. So we were the consortium lead that achieved the first FDA recognized use of hemp seed meal in laying hens and the AFCO approved ingredient definition of hemp seed meal. So this was extensive and expensive work, especially that first application. We've developed a great working relationship with those entities now. So subsequent applications are going to be much more expedient. And we're actually next on our list for submission is the consideration of a hemp seed meal and ruminants. So this is a very timely presentation. We're excited to hear. Just want to call out lastly and thank our existing members. Thank you, thank you. We appreciate your support. What we do is costly, so we ask the industry to join. We have leveled memberships, other ways you can show your support, including in-kind donations. But our biggest need, of course, is cash, and we use those funds to pay our wonderful application scientist that creates the white papers, et cetera, who is your host today, Christine, as well as for any additional little error trials that FDA calls for. So please consider supporting, become a member, go to hempfeedcoalition.org. Again, we're a not-for-profit, so all donations are tax deductible. And that's our spiel. Take it away, Christine.
All right. Thanks so much, Wendy. I'm always so excited when you're here because you do a way better job than I could at that spiel.
I care.
Oh, all right. So just a little housekeeping. If you guys have questions today during the presentation, please enter them into the Q&A section that should be up above on your app, and we will address those at the end of the presentation. All right. I put a little note in there, so hopefully you can find it. Now on to the main event. Today's speaker is Dr. Kimberly Wellmann. Dr. Wellmann grew up with a passion for large animals and completed her undergraduate degree in animal science with a minor in business administration at Texas A&M University. She earned her Master of Science in Agriculture from Tarleton State University and completed her PhD at Texas Tech, where her research focused on nutritional factors affecting muscle development in feedlot steers. Following her doctoral studies, Dr. Wellmann worked as a sales manager and calf consultant for American Calf Products and later with a beef cattle producer in Anton, Texas. She is currently an assistant professor of animal science and beef cattle specialist at Tarleton State University. Notice how she stayed in Texas but has made the rounds. We were talking about that before the presentations. Her research focuses on feed additives and micronutrients and their effects on growth, performance, reproduction, and muscle development in beef cattle. We're very excited to have Dr. Wellmann join us today. So please join me in welcoming her as she presents. I had a different title, but the effective dietary hempseed meal on growth, performance, feed efficiency, and blood parameters in yearling rough stock bulls.
I appreciate the introduction. I had a bit of a title change because I didn't want to steer too far away from what my master's student originally produced. She was kind enough to actually let me modify her thesis defense slide. So that's actually what I'm presenting today. And this is directly correlated to the actual publication, which is linked there. So this study was originally developed in conjunction with Dr. Kimberly Gay and Dr. T. Wayne Shortner here at Tarleton, as well as a couple industry collaborators. And that's what allowed us to do this. So before I get into the actual project itself, I'll talk a little bit about the background of, why we did the project, what's a hemp plant, what hemp seed meal is, some of the legislation surrounding this and why we ended up getting into this. So some of this is going to be a bit outdated information for those of you that have been on the forefront of, really advocating for the use of hemp seed meal and hemp byproducts as a part of the animal feeding industry. But it's always good to review information because we learn through repetition. So when we look at industrial hemp, we're talking particularly about any parts of the cannabis sativa plant that have less than a 0.3% THC content. And the derivatives that we're going to use or talk about most are hemp seed meal, hemp seed cake, and hemp seed oil. Most people are fairly familiar with hemp oil and hemp fiber. But when we extract those portions of the plant, it leaves us with this portion of the plant that has a pretty interesting nutrient profile that allows us to fill in some gaps in animal diets. And we really hadn't studied it extensively in the ruminant diet, which is how we kind of got into this. So looking at the byproducts of hemp oil extraction, really the things that we're going to be interested in are the actual nutrient content or that profile, and then the potential for sustainability. So the idea of the nutrient content is that it really has this more complete nutrient profile where we see things that include fiber, fat, some starch, a considerable amount of protein, and there it's a pretty unique individual ingredient in terms of animal feeding because there's not very many other byproduct meals that have a similar profile. Usually, they're high in protein, but not very high in fat and fiber, things like that. And then in terms of the sustainability component, obviously, like was stated earlier, when we look at agriculture, we're really interested in utilizing every part of the plant out of the crops that we harvest. And hemp seed meal is an example of doing that. So when we talk about it in terms of legislation, you guys, like I said, are probably fairly familiar with this idea of the Hemp Farming Act, which is great because that legalized industrial hemp production in the US. But at that time, it was not authorized for use in feed or in animal feedstuffs. Particularly, it was not grass, meaning it was not generally recognized as safe for a feed ingredient. And so we got into a discussion with AFCO, and by we, I mean you guys, the Hemp Feed Coalition, got into a conversation with AFCO, as well as the FDA, looking at different guidelines regarding these. And there was actually a call to action letter at the beginning of the year that we did this project, where we're talking about the concerns of the potential inclusion of hemp and hemp byproducts as feed ingredients. Fast forward a couple of years later, after, you know, a substantial amount of research, including this project that I'm about to go through, and a couple of other projects that Dr. Gay was involved with, we get to January 2024, and there was an approval vote after a considerable amount of discussion about the amount of cannabidiol or CBD itself that was allowed to be in it on a parts per million basis. However, big thing there is that it got approved. Okay. So when we look at as a feedstuff, like I said earlier, hemp seed meal and the hemp plant as a whole are high in fiber, fat, and protein. So just for those of you who are not super familiar with a lot of feed and byproduct feeding, most byproduct feeding is designed to incorporate protein into animal diets. And usually those come from oilseed meals that are not super high in fiber on the byproducts end of the diet. So the fact that we have this high protein, high fat, potentially high fat, depending on how it's processed, byproduct feed that's still high in fiber, it was really what drove the interest of using this product as part of the ruminant diet, because ruminant animals handle fiber digestion much better than non-ruminant animals. And so when we look at that compared to some of the other things that we might use to suit this need, it's going to be much more calorically dense because of the fat content. That being said, when we consider ruminant needs, we do have to be a bit careful when it comes to fat inclusion, which you'll see dictated the inclusion of the diet when we talk about this project. But we have to be a bit careful because of the relationship with fat and the microbes in the ruminant animal. Okay. And we'll talk more about ruminant animals specifically for those of you that may not be super comfortable and just be able to rattle off what a beef cattle GI tract looks like. So no stress if that's what you're already thinking. What is this word ruminant? Okay. So when we look at it as a potential feed ingredient from more of the industry, less of the nutrition side, it does seem to be pretty cost competitive to our base supplements, things like soybean meal, cottonseed meal, etc., depending on the producer and the intent behind whatever they're pressing it for, usually oil. And so the idea of not wasting this portion of the plant can be really beneficial from a sustainability component, marketability component, and those sorts of things. There was another 2022 study that observed the effects of hemp seed cake. So cake versus meal. Cake is generally the compressed version of hemp seed meal. Maybe it's been extruded in a slightly different way or has been heat treated in a slightly different way. Could also potentially have a binder. It kind of depends on the type of production when we talk about cake. But essentially it's those like compressed cubes or the giant pellet looking things when we talk about hemp seed cake. But generally speaking, the comparison between that and the other, the control diet, they didn't see any significant effects on average daily gain or efficiency gain, and body fat and conformation were also unaffected. Now, you might be like, well, Dr. Wellmann, why did you go ahead and proceed with the study if, you know, there's already a study out there that said there wasn't any difference? Okay. What people have to remember when we talk about studies that produce no difference between a control diet and whatever the treatment diet is, oftentimes that ends up being a really good thing for whatever the treatment diet is, because we want it to be competitive on the same level. We don't want to see a drop in the performance of that animal. But we also want to make sure that it's going to not create any unintentional side effects, whether that be behaviorally or otherwise, because that can be indicative of super nutritional impacts that may end up putting that product under other regulation, control, or legislation. Okay, so just kind of getting into the hemp plant itself. It's one of the oldest domesticated crops, and it has 4 distinct parts. When we look at it, we're thinking, okay, seeds, these are a good source of nutrition, sustainable fuel. They're really high in fat, calorically dense. Okay, the stalks in particular are very high in fiber. The roots can often be used for medicinal purposes. And then the leaves often contain CBD and omega-3s. Okay, so just some things there to consider whenever we talk about the portion of the plant that we're going to use. And this would incorporate portions of the leaves and stalks predominantly when we talk about hemp seed meal to some extent, but also the primary portion is the residual of crushing those seeds for oil production. Okay, so when we look at whole hemp seeds, okay, again, that seed portion, we're looking at a pretty even spread of the nutrient profile, where we have 25 to 35% of that, including lipids. So about 1/3 of it is fat or oil. About 1/4 of it is protein. Okay, another 1/4 of it is going to be carbs. And of those carbohydrates, about 98% of the carbs included with the whole hemp seed are referring to the seed hull, and it's going to contain dietary fiber. Again, that's something that I like to note when we're talking about feeding these products to ruminant animals as well as hindgut fermenters, because they have a greater capacity to utilize fiber. And then 4 to 7% of that seed is going to be ash. For those of you that don't know, ash is going to generally refer to the mineral fraction of that seed, things like calcium, and any kind of like salt electrolytes, those sorts of things, because they don't burn off the same way that our general macronutrient classes do. Okay. Now, one major factor here that we have to consider is that the method of processing will impact the nutritional content of the hemp seed derivatives, particularly hemp seed meal, because roasting, seed hull removal, and oil extraction all impact those classes of nutrients that are listed above. Okay, example of that being roasting, for those of you who didn't know, or cooking something, can directly impact the folds of the proteins. It also generally removes any water fraction. It can cause a browning reaction where you can have carbohydrates that essentially stick to proteins, and that's called a Maillard reaction. And those kinds of things can impact the digestibility or the ability of the animal to actually digest, absorb it, and utilize whatever that class of nutrient is that's in. Seed hull removal would obviously most directly impact the fiber content or the carbohydrate content of that product. And then oil extraction, depending on whether it's cold pressed or a solvent is used, directly impacts the calorie content or the energetics of whatever product that is going to be used, because the more oil that's going to be pulled out, the lower fat the byproduct is, therefore the lower the caloric capacity for whatever that byproduct is. Okay, so when we look at cold pressing, this has become the more common and more conventional method of oil extraction. It's ideal for pulling oil out in its most pure form, and it only has about a yield of 27 to 31%, so a little less than 1/3 of what it could potentially be relative to the amount of oil in the plant. Okay, so or in the seed. And so this is going to have the least impact on the protein, that browning reaction, that kind of thing, because you're not going to heat that seed up to a point where it could potentially burn the byproduct component. of this hemp seed meal production or hemp oil extraction. And so it doesn't get over 48 degrees Celsius. So that's around 120 degrees Fahrenheit, a little bit more than 120, between 120 and 150 degrees, I think, if I recall correctly. And then maintaining the moisture during this pressing method is also really important. Okay, the flip side of this is going to be solvent extraction. And so really common solvents that are used to pull oil or lipophilic compounds out are going to be ethanol, which is an alcohol, or hexane. And it is a more complicated process, but it is highly efficient. And so you can actually see an increase in oil yield of, anywhere from 9% down to about 5%. And so this would maximize the amount of oil that we're able to pull out. But again, it's not going to pull it out in the purest form. And the solvents used could impact the quality, the nutrient profile of the hemp seed meal later on down the road. So when we look at ruminant animals specifically, okay, I'm just going to give a brief overview here, and we'll see some pictures here in a second that might be a little bit helpful in better understanding the ruminant GI tract. But as human beings, when we consume food, we chew it up, so we masticate it, we insalivate it, it gets moist, we swallow it in a bolus form, it goes down our esophagus directly into our stomach. So for us, it's not going to be acted upon by any kind of fermentative microbes. And then that stomach is our gastric organ. And then later on down our GI tract, it's going to be acted upon by the enzymes in our small intestine. And potentially, depending on the development of your personal microbiome, some fiber will be digested a little bit by the microbiome we have in our large intestine. Okay. When we talk about horses and rabbits, those are hindgut fermenters. So the front part of their GI tract, or their foregut, so everything prior to their large intestine, is going to act very similar. And then when we get to the hindgut, so the large intestine, which is going to be the cecum, small colon, and large colon, those are going to be the portions that contain this large microbial population. Okay, so this is why horses can graze on grass and eat hay and actually get some kind of energetic benefit out of it. Whereas like us, if we eat grass or a salad or something like that, we don't utilize calories out of that hardly at all by comparison to something that we can digest in our small intestine. Ruminants are different. So they're a little bit flip-flopped, or they kind of have these end cap of microbe containing organs. So when a ruminant animal swallows its feed stuff, it goes down their two-way esophagus, intentionally two-way esophagus, and it hangs out in the rumen and the reticulum, which are these big fluid-filled chambers that host a variety of microbes that are very good at digesting fiber, starches, sugars, proteins, etc., and don't handle fat, particularly unsaturated fat, very well. But this allows that long stem roughage to hang out in there, be digested a little bit. They can also ruminate, which is where they regurgitate their feed stuff into their mouth, re-chew it, decrease that particle size, re-swallow it, let it ferment some more. And as those particles become saturated and become smaller, they'll filter their way down through the rest of the ruminant GI tract. And so that's going to then go into the omasum, where that feed stuff is dehydrated by that omasum. All the water is squeezed out of it before it goes to their gastric organ, which is the abomasum. So that's the closest thing to their stomach. And that's where you start seeing that protein digestion through acid enzyme hydrolysis, and then into their small intestine. We'll have more enzymatic traditional breakdown of different feedstuffs. Now, I gave that rundown. It's pretty quick. I didn't put a picture up here because I'm just that mean professor that expects you to listen and know the things. But from there, the rest of their GI tract is much more like a non-ruminant or like a human once you get to the small intestine. The key about this big fermentative vat is that these microbes that are able to break down fiber produce volatile fatty acids, which is what you see here, abbreviated by the term VFA. So volatile fatty acids can be passively absorbed into the bloodstream and then converted into different forms of energy based on what the animal's needs are. So for us, for non-ruminants, we just eat sugar, we eat a candy bar, whatever, we have high blood sugar. You can help directly support your central nervous system and its need for glucose by just consuming glucose directly. Ruminants rely on something called gluconeogenesis as a great portion of maintaining their central nervous system because they predominantly absorb volatile fatty acids. And then their liver does a great job of converting propionate and other VFAs into glucose after it's been metabolized. Okay. So a lot of information I just threw at you, but for those of you that are not familiar with cows, hopefully that gives you a bit more background. So when we look at micro and macronutrient utilization, so we're in the macros right now, we kind of started with protein and how protein can be hydrolyzed into amino acids, which are just the building blocks of protein. And then they can also be transaminated or depending on the needs of the microbial cells in that ruminant animal. And later that's allowed to either contribute to microbial cell protein. So those microbes in the rumen get flushed down the GI tract as well. And the microbes themselves get broken down as a source of protein, actually about 60% of the protein that animal is able to utilize. And then there's also the energy component, which is generally associated with VFA production. Okay, so a lot of information there. But the general takeaway here is from a protein standpoint, ruminants don't really care about the amino acid profile because the microbes are going to just turn it into whatever amino acids they need. And then that ruminant animal is going to digest and absorb the broken down microbial cells as its primary protein source. Okay, so this is going to look something like this. I'm going to do a really bad drawing here really quick, and we're going to put our head of our cow right here. Okay, so this is the head end of that cow. Okay, and this is the tail end of that cow, which should be pretty obvious based on the pile of feces right there. Okay, but this just kind of looks at this process of protein digestion in cattle. So when we say true protein, we're talking about complete protein. That's going to be the protein derived from plant products. This is not something that is synthetic. It's not amino acids specifically, and it's not non-protein nitrogen, which you'll also see that fraction of non-protein nitrogen right there as well. Okay, and so that's this fraction. We try to reduce the percentage of non-protein nitrogen that we feed them because it can end up essentially going to waste if you feed too much non-protein nitrogen without sufficient fermentable carbohydrates to help those bacteria facilitate microbial cell protein production or just their own reproduction of bacteria. Okay, so that's going to go through generally about 60% of that ends up being utilized and or developed into microbial cell protein. There's a whole urea cycle going on here that incorporates ammonia overflow that may be excreted out through the urine or recycled through the liver and put back into the saliva, those kinds of things. And then you'll also have escape protein or rumen undegradable protein that ends up making it directly to the small intestine intact the way it was consumed. Okay, but that's kind of the takeaway there. Okay, we also have to consider carbohydrates as a primary macronutrient for ruminants. Most livestock animals should have the majority of their energy coming from carbohydrates, okay, as opposed to fat, because they all evolved into animals that utilize either structural or non-structural carbohydrates as their primary substrate for energy in some capacity. So when we look at high energy and energy sources for ruminants, the majority of them contain 40+% carbohydrates. Okay, 40%. There are a couple control diets or conventionally confined feeding diets that contain as low as 20% carbohydrates. I would tell you that that's very uncommon. And the majority of these are going to be structural based. Okay, so for dairy cattle specifically, we want to cap our non-structural carbohydrates around the 30% range, but then still allow for structural carbohydrates. What do I mean by structural and non-structural? Structural is going to be like fiber. Okay, it's going to be those carbohydrates that are linked sugars by beta bonds, as opposed to non-structural, which predominantly are sugars that are bound together by alpha bonds. So that's fiber versus starch, essentially. It'd be the idea of like lettuce versus a potato. Okay. When we talk about microorganisms that are going to attach to these feed particles, we have specific ones that are amylolytic, so starch-loving microorganisms, as opposed to cellulolytic or fibrolytic that would prefer to attach to cellulose and hemicellulose, things like that. Okay, and then essentially that carbohydrate is going to be hydrolyzed into smaller di and monosaccharide components. Then you can have sugar fermentation, and as that bacteria, predominantly bacteria, utilizes that carbohydrate, those bacteria have their own output that is a byproduct of that bacterial metabolism of that carbohydrate, and they will produce acetate, butyrate, and propionate in usually about a 50 to 60 to 10 to 30 to 40 ratio of acetate, butyrate, and propionate. Okay. And then these are just very small fatty acids. So they are 2, 4, and 3 carbon relative to the way they're written on the slide here. And they are used by that animal directly for energy. Okay. Then we get into lipids. So this is our third class of macronutrient, and the utilization of lipids in most mammals is going to be through this process of lipolysis, where we have the breakdown of the lipid by hydrolysis, and that's going to allow the animal to utilize it. So, or break it down enough that they can absorb it and store it. The actual utilization of fatty acids for energy goes through a process called beta oxidation, where you get into a cell, you have enough oxygen, and it chops 2 little carbons off of a fatty acid chain at a time. And chopping off that carbon, that reaction is what produces energy. Ruminants specifically have this very unique ability to create saturated fats. Okay, so when we talk about a fatty acid, okay, to be the chemistry nerd for a brief moment, we've got a carboxyl group, and then we've got our fatty acid chain. Okay, so that carboxyl group is the foundation of most fatty acids. But that carbon chain, okay, each carbon wants to have 4 bonds. Okay, each of those bonds needs something on it to stabilize it. So if we have a double bond from carbon to carbon, we don't have hydrogens filling that void. If we only have single bonds in a carbon chain, each carbon is going to have 2 hydrogens coming off of it to utilize the extra bond space that a carbon atom has to have to be stable, essentially. Okay, so unsaturated fats are those that contain double bonds, because that fatty acid chain is not completely saturated with hydrogen. Okay, that's where the term comes from. Okay, and unsaturated fats are bendy in shape, and so they don't sit together as tightly, which is why they generally are in the form of an oil that is some kind of liquid when we talk about unsaturated fats. Okay, saturated fats have really nice straight carbon chains with hydrogens on both sides, and they stack together really densely, which is why most of them are the ones that are solid at room temperature. Okay, then you got the in-between, which is trans fats. And those trans fats usually have double bonds in them, so they are unsaturated in some capacity, but the molecule has twisted so that the double bond ends up making the molecule more straight, and so then they again pack together more densely, and they function in the body more as a saturated fat than they do as an unsaturated fat, which is why all of the healthy people in the world get real stressed about them, because they kind of trick your body. Okay. When we talk about absorption of fatty acids, particularly medium and long chain fatty acids here, so we're not just talking about our VFAs. So these are going to all be 6+ carbon in length when we get into how these guys are absorbed. Those shorter chain ones will absorb directly through the cell membrane. But most medium and long chain fatty acids require micelles in order to be absorbed into the enterocytes, or those intestinal cells, because they the GI tract of all mammalian species is an aqueous environment. It's water-based, and as you guys have all experienced, fat and water don't mix. Okay, and so we've got to find a way to cheat and make them mix, and we do that through micelle production, which is a water-loving coating on the outside and a fat-loving coating on the inside. And it's this little ball that's allowed to be transported into those enterocytes. Okay. Once it goes through that, they're put into the lymphatic system, occasionally the bloodstream, predominantly the lymphatic system, and then they go to the liver and get distributed out. Okay. So here's a better picture of what the GI tract of a cow looks like. So we've got our esophagus into our rumen. Okay, that's the big gray area. This little sac up front here would be the reticulum. And then eventually it's going to be impacted by VFAs or microbial digestion and converted into VFAs, or we'll have undigested lipids that are converted into more saturated fats because of that process of biohydrogenation, where those microbes go and stick hydrogens onto that carbon chain. And then that will go into the rest of the GI tract and be absorbed through those micelles into the lymphatic system, and then go contribute to things like milk fat, subcutaneous fat, and intramuscular fat. Okay. So we also had to consider a variety of different studies before we started our own study. And so we predominantly focused on ruminant-based studies. So we had several small ruminant studies that had been done, sheep and goat studies. So we started there, where we predominantly didn't see a ton of difference in dry matter intake, body weight, or gain in the 2021 study. But in the sheep that were fed the hemp inclusion at the greater rate, had greater dry matter, organic matter, and protein digestibility, which is really cool. That means that diet was more digestible and better able to be utilized by those animals compared to the low inclusion diet. And then in terms of NDF and ADF, again, that digestibility was lower in the control sheep, meaning that the fiber digestibility in those sheep that were fed hemp had greater fiber digestibility. That's great. Okay, in terms of utilizing a new feed ingredient in a diet. When we looked at the 2011 study, there was a greater intake and crude protein intake for lambs that were given the hemp cake as well as the peas, okay, as opposed to rapeseed cake or canola cake, is probably what most of you are more familiar with. Rapeseed and canola are the same product. But basically, when they looked at the final weight, okay, the canola and the peas actually performed better. So it depended on what the diet was between these two studies and when it was done and how it was managed as to whether or not the hemp cake or hemp seed meal performed better than the control type diets. Okay. We also had this hemp seed meal fed to goats in 21 and 22. And there was very little effect of hemp seed meal on the carcass weight or dressing percentages compared to the control diets. And so up to 30% in that first boar goat study there, the carcass traits were very similar. Like up to 30%, the hemp seed meal performed just like any other normal feed. Okay, and again, like I said, when we're talking about including feedstuffs in animal diets, it performing at the same level as whatever the industry standard is usually really good for that product. When we get into the 2021 study here, the big takeaway is that the fiber intake, so neutral detergent fiber and acid detergent fiber intake increased with the hemp seed meal supplementation. If you combine that with the ability to have greater fiber digestibility, that's a really good sign from an energetic standpoint. Okay. Hemp has also been fed to dairy cattle. So in 2010 and 2022, there were a couple of dairy cattle studies, one that looked at hemp cake compared to a traditional dairy diet, and the other that tried to compare it directly to soybean meal. So when we looked at hemp cake in the 2010 study, it did impact energy corrected milk yield, where the hemp cake actually improved some of the yield, but decreased milk protein and fat levels. Okay, and the milk had a greater urea concentration, which will become a point of discussion later on after we go through the current study. Okay, as far as hemp cake versus soybean meal supplementation, in the Simmental dairy cattle, hemp cake supplementation didn't impact average daily gain or the feed efficiency of that animal. And so generally at the low inclusion level, it served as a sufficient replacement for soybean meal. Okay, for those of you who don't know, soybean meal is like the gold standard when we talk about a byproduct-based protein supplement for most livestock diets because of its amino acid profile. That being said, generally speaking, our ruminant animals don't have the same amino acid profile demands that non-ruminant animals do. So ideally, even if a hemp cake or hemp seed meal type product doesn't have the same profile that we need for non-ruminant animals, where it's really high in lysine, etc., it could really serve as a strong option for supplementation in ruminant animals. Okay, and then of course we looked around for studies associated with beef cattle. Mind you, this one was actually published after our study was run, but still served as a good comparison because it was run very close to the same time that we ran our study. And so when we looked at the steers on the hemp diet in the 2023 published study that ran it at a very similar inclusion level to us, we saw an increase in organic matter intake, but a decrease in total tract digestibility, particularly nitrogen digestibility. And nitrogen utilization is pretty important whenever we talk about the protein deposition into muscle going into feedlot animals. Okay, so pretty good that we saw the increase in nitrogen digestibility for that hemp cake compared to the decreased control diet. And then in terms of the 2022 study here, we didn't really see differences in final body weight or average daily gain for veal-based or veal production calves. And then there's actually an improvement in dressing percent and carcass conformation, so the ratio of bone to muscle to fat for calves that were fed the hemp seed cake, okay, or hemp cake. And so both of these studies and their positive results really supported the utilization of hemp byproducts as a portion of the diet in beef cattle. Okay, so that's going to lead us into the study that we actually ran with the objective of evaluating the effect of dietary hemp seed meal inclusion on growth performance, feed efficiency, and blood metabolites in yearling rough stock bulls. Okay, so looking at these guys, we had 40 head of rough stock bulls that were supplied by a private stock contractor. We used rough stock bulls because the idea is that these animals were not going to go back into the food chain, which was very, very necessary at the time because this was in the heat of that call to action by AFCO. And so we fed them in 4 head pens because statistically speaking, we needed to average the animals in the pen and feed by pen in order to have a strong enough study to evaluate a diet and also not run into issues with pseudo replication. So we had 5 pens that were fed hemp seed meal as one of their supplements versus 5 pens that had a control supplement developed for them to try to mimic the idea of hemp seed meal. Okay, so when we look at the treatments in this, okay, rather than trying to do a direct comparison to another common feed ingredient, I actually formulated a supplement, a control supplement, using soybean hulls, cottonseed meal, and an added fat to mimic the hemp seed meal that we actually got. So we actually did a nutrient profile analysis on the hemp seed meal that we were using, and then I formulated something comparable using very common supplements and feed ingredients for the livestock industry. The reason I did this is because rather than just looking at the macronutrient differences in terms of it being a caloric difference or a protein content difference, I wanted to say, actually, what if it's specific to the type of fat, not just the amount of fat? What if it's specific to the type of protein, not just the amount of protein? What if it's specific to the type of carbohydrate, not just the amount of carbohydrate found in these supplements? And so these were designed originally to be as close to isonitrogenous and isocaloric as they could be. You'll actually see that there are some slight differences here where the control supplement ended up being a little bit more calorically more dense, but it was on such a small scale compared to just straight up trying to use cottonseed meal or soybean meal or something like that. From an ingredient standpoint, if you look at other byproducts out there, sunflower meal is the next closest product to hemp seed meal, just for reference, in terms of macronutrients. So these cattle had an acclimation period where they were just fed the basal diet without the added supplement. So it was the Stocker 10. So as a portion of the concentrate part of the diet, they had just a normal base Stocker 10 feed. And then another 20% of the concentrate part was the supplement. And then they were fed 50% of their dry matter intake as Bermuda grass hay, which is really common for cattle in the growing stage to be kept on some kind of hay. Okay, they were fed twice a day at 7 a.m. and 5 p.m. In order to record intake, orts, or feed refusals were recorded daily. Generally speaking, they used slick monk management, which is where we were trying to feed them ad libitum in order to see who wanted to eat more, essentially. But it was adjusted at a ½ pound per head per day if they slicked their bunk and needed more feed. Okay, we collected all of our samples on day 0, 21, 42, and 63. It's a fairly short study because the majority of it was internally funded and it had a tight timeline for when deliverables needed to be put out. We calculated dry matter intake, average daily gain, and then we used whole blood samples fractioned out to a serum and plasma, and then those were analyzed via VET scan and a couple other analyses. Okay, so this just gives an idea of what we were looking at in terms of the timeline for that study. And there was a month-long or a 4-week adjustment period so that those cattle were well acclimated to the pens and feeding practices that we used during the study. Okay, so from a statistical standpoint, we used the Glimmick's procedure of SAS 9.4, where pen was the experimental unit, again, because we were feeding by pen. And we looked at the main effects of treatment and day. And then anything that was considered significant had to be a P of less than 0.05. And tendencies were discussed between 0.05 and 0.1. Okay, so just take a second to orient you to these figures. Most of my tables will be something comparable to this, where we will have whatever our dependent variable is along here. And if it's broken out into time, it will also be on the left columns. Across the top, you'll see the column titles, including the treatments, so control and hempseed meal diets. And then we'll have the standard error of the mean and the p-value for each of those different dependent variables. Okay, and so this shows you initially there was less than a 5-pound difference between groups, and same thing at their final body weight, there was less than a 5-pound difference between groups, and they were not statistically different at either time point. Okay, so over here looking at our p-values, you'll see often that we don't have a significant p-value, meaning that the treatment versus the control, so the hempseed meal versus the control is not different. That being said, when we looked at the first 21 days of actually putting them on this feed, remember they had been acclimated together, learning how to eat this feed beforehand. So even within that 0 to 21 day period, we actually saw a difference in average daily gain and feed efficiency, where our hempseed meal animals were actually requiring a bit more feed. So they were eating more, okay, and not gaining much more at that time. So those are tendencies that we saw there, which was really interesting. But if you look at that in terms of dry matter intake, so the amount that they were actually consuming, the fascinating part about that is that they actually were consuming more. And oftentimes, especially in higher inclusion studies, we would see them kind of back off of hempseed meal from a palatability standpoint. But these bulls ate it like champs. Okay, they really enjoyed it. And then you just see slight differences over time of, those hempseed meal animals were really consistent at eating about 8 kilograms per head per day. Whereas the dry matter intake animals increase their intake with their body size. Okay, so same thing here, we'll have blood metabolite tables where the specific metabolite is on the left column. And then we'll have our treatments up here, but they're broken out into columns by day. And that allows us to better visualize some of the specifics here. Okay, for the purposes of time, I actually am not going to talk too much about what's going on here unless there's some kind of treatment tendency or difference. So we'll talk about the albumin being tending to be different for treatment and being different by day. But it's really common to see a change in blood metabolites for all animals over time as they mature, because the way that they utilize nutrients as they become more fat changes their blood metabolites. Okay, when we look at these other parameters, again, I have some bolded interactions and bolded main effects here that we could talk about. But the reality is for the purposes of this talk, I did not focus very much on day effects. I really only tried to focus on interactions and treatment effects, because again, most of the day effects are going to have to do with the animal's physiology as they mature, as opposed to things like globulin, which is one of the primary immune proteins in the blood. This is what allows us to know if an animal is going to respond to some kind of immune challenge, essentially, and if they'll have the abilities to build antibodies. And this was different between treatments, where we actually had an increased globulin content of grams per deciliter globulin for the hemp seed meal animals compared to the control. Okay, and to better visualize that, we're looking at something like this, okay, where we had a little over 5 grams per deciliter of globulin, as opposed to the control diet, which only produced 4.4 grams per deciliter of globulin protein. Okay, the other thing here is that you'll see a big jump of their globulin content as they went on to the diet. And again, they had all been acclimated together up to day 0. And so the change in the feed by adding this higher calorie diet or supplement to the diet actually did create a pretty big jump, and then it plateaus for the remainder of the feeding. Okay. We also saw a treatment tendency to impact glucose, which was another thing that was kind of interesting, where we actually had lower glucose circulation in hemp seed meal animals. So the hemp seed meal diet tended to produce animals that had a lower circulating glucose content. Again, really fascinating there. We've got some theories as to why, and this is going to be comparable to the impact that it also had on albumin, which was on that first slide that we chatted about. Okay, we also saw an interaction for potassium and total protein. And I have theories on both of these. Predominantly the fact that it has to do with the temperature outside. So for those of you who don't know, this study was run from July through October, basically. Not quite October, but essentially October, which if any of you have been to Texas, it's pretty warm here. We're supposed to hit 98 today, and we're still in June right now. And this year in particular, I think we went 40 days with no measurable quantities of rain. And I think we had 20, maybe not, maybe we had a dozen days in a row where we were hitting triple digits every day. Okay, so these are definitely heat stress conditions. Now, these animals were housed in covered pens with dirt floors, and they had access to fans. And so while that does help, it does not mitigate the utilization of different electrolytes, things like potassium utilization in muscle, and it does not negate the potential for dehydration. Okay, so dehydration can come into play here. And so in general, we saw slightly higher globulin content in the hemp seed meal animals compared to the control, depending on the day. Okay, so the last 3 days, it was higher as opposed to the first day where it was lower. So they kind of flip-flopped total protein content. And then those potassium levels, we see something similar where we have some overlap on the 21st and 42nd day, but very similar by the last day on the potassium, as opposed to where they started out, where our control cattle had much greater potassium per liter of blood. Okay, so that's going to look like this. Okay, basically they started out different and became the same. Okay, and then here, my estimation is that at day 21 and 42, we had the greatest dehydration in the animals for this total protein. But with that in mind, hemp seed meal did a better job at maintaining the total protein, which is also directly related to the globulin that we talked about earlier. Okay, now talking about total protein gets directly correlated to serum urea nitrogen as well. Okay, and so this, we did see a difference over time, but more interestingly was the main effect of treatment here, where we actually see that the serum urea nitrogen for hemp seed meal consuming cattle is slightly higher than control cattle. Now, this will tie back directly into that conversation I brought up in the literature review where our dairy cattle showed greater milk urea nitrogen. So there is definitely something, for those of you who don't know, urea nitrogen in the blood or in the milk indicates that the animal may not be utilizing that protein source as effectively as they could be, okay, or they have a surplus of protein in some capacity. And so they're recycling the nitrogen in their system. And so this to me says, okay, either there's something that they're not able to utilize out of the protein source in that hemp seed meal, or it needs to be fed in conjunction with more soluble carbohydrates for that animal to be able to better utilize the nitrogen for microbial cell protein development. Okay. We also, just kind of to like wrap up in this study, did not see an impact of body weight or average daily gain. Okay, so that's a summary of that first table. We saw some slight differences at that initial feeding period, but in general, interim DMI was not impacted. Okay, we did see greater serum urea and nitrogen concentrations for the hemp seed meal fed cattle. And then over time, okay, we saw increases of body weight, serum urea, nitrogen, and dry matter intake as associated with what we'd expect as cattle mature. And then we've got this decrease in average daily gain over time, which is also normal for cattle as they mature. Okay. This just wraps up the same blood metabolites that we just kind of talked through. So we had an interaction for potassium and total protein. Okay, we saw lower glucose and albumin in hemp seed fed bulls. And then we saw greater globulin levels in hemp seed fed bulls as well. Okay, and then again, slight differences associated with maturation and the temperature outside over time. Okay, and so when we go back and compare this performance to other studies, okay, really we saw that there was similar digestibility and total digestible nutrients between the two diets, which is good because they were formulated that way and there were no major red flags from either diet. Okay, so generally speaking, with that in mind, we would assume that those differences are based on the hemp cake or hemp seed type composition. Okay, and our study was actually very similar to some of these other studies that really didn't see a major difference in performance, which again is beneficial because that makes the hemp seed product one single product that I can replace potentially 3 different products with, and it's still going to perform in the same way. Okay. There is definitely something that should be further investigated regarding plasma and serum urea nitrogen or milk urea nitrogen, because there is something that increases the urea cycle in most ruminant animals that have been fed some kind of hemp byproduct based on the increase in urea nitrogen levels in a variety of different studies. Okay. Our study was unique in finding that there was a tendency for a glucose difference in hemp-fed animals having a decrease in glucose circulation. But the reality of that is because our ruminants don't rely on postprandial glucose and insulin spikes, I expect that it actually allows them to better have some kind of glycemic control. So it serves as maybe a bit of a modulator for blood glucose. And then as far as some of the other metabolites, there were not a ton of other studies that looked at these kinds of differences. The goat study had a lower serum total protein, which really conflicted with our study. But again, it depends on the time of year. There are other confounding factors that can potentially impact some of these. One of the number one being dehydration, and then reactivity to the environment. Okay, so to kind of just wrap things up here, basically, hemp seed meal as a singular product has comparable nutrient availability to a supplement that was formulated to mimic the macronutrient profile of hemp seed meal. The difference is that I can use hemp seed meal as a singular ingredient as opposed to somewhere else where I'm going to have to use potentially 3 ingredients to create the same nutrient profile. Okay. I'd like to really thank my PhD candidate. She actually is going to defend here in the next 2 weeks her PhD, but Samantha McGovern, she was the master's student on this project. Dr. Kimberly Gay and T. Wayne Shortner were both vital in the funding acquisition portion of this study. And then we had some notable mentions, which were recently awarded Dr. Ryan Springer, and then Drs. Brady and Dr. Runyon, as well as Lane Smith, who was the counterpart master's student for the behavioral portion of this study. Okay, with that in mind, I will happily answer any questions that you guys have. And again, this is these data are published in the article that's linked here.
Well, thank you so much, Dr. Wellmann. That was very thorough. Apologies. For the sake of time, I want to address the one question we have right now in the Q&A, and I think that Dr. Wellmann would probably welcome any other follow-up questions you might have via email, I would assume. But if you could summarize, maybe recap the work you just did, what makes hemp seed so special?
So hemp seed meal, particularly, I think that what makes it special is that I can use one product to essentially replace 3 products. So the supplement that I produce that's so comparable to that profile, it requires me to have something that is high in protein. So I used cottonseed meal for that because it had a better fiber content than an alternative like soybean meal or canola meal. And then I use soy hulls to help up the fiber to be comparable to hemp seed meal. And then I had to go and use essentium, which is an added fat, in order to get it to the fat level that the hemp seed meal was at. I had to use a considerable portion. So that's 3 separate products that can be quite expensive, whereas hemp seed meal just is its own product. And depending on how it's processed, so however much fat is left in it, you can really change the product itself in a positive way for whatever animal you're feeding it to. So for cattle, I might want something that has a lower fat hemp seed meal, whereas for, pigs or chickens or something like that, maybe I leave in more of that fat to get better caloric intake. But yeah, hemp seed meal special because it's basically 3 products in one.
That's a really, I think, great way to wrap up the main message here. So like I said, I'm sure if y'all have additional follow-up questions, Dr. Wellmann would be glad to answer those via email. You can just reach out to the HFC team or to her directly, and we'll get you connected. But for the sake of time, being a little bit over, I am gonna just thank you all for being here with us today. Dr. Wellmann, thank you so much for your time and sharing your hopefully soon to be PhD students, or I guess newly minted doctor.
Hopefully, yeah, newly minted doctor.
Yes, I will hope for that as well. But thank you so much for being here with us. Just a real quick wrapping up. I just want to remind you all that our next Hempseed Coalition webinar will be Thursday, July 16th. Mark your calendars. And that will be at 10 a.m. Eastern Time. We will be hearing from Dr. Ruggero Amato again, and he will be discussing hemp extracts in rabbits and dogs and the effects on stress and quality of the life. So you guys can see that on the main screen. Thank you again so much for being with us. I hope you all have a great rest of your day, and we will see you next month.
Have a good one. Thank you so much.
Thank you.